everyone, welcome back to my channel. This is lecture 4 of chapter Structure of Chromosomes, Cell Cycle and Cell Division. And in today's lecture, we are going to study about cell cycle. So we will study cell cycle in detail, okay? And a brief introduction about phases of mitosis. Okay, so let's begin. I am drawing a very simple diagram, and through this you will be able to understand cell cycle very easily. Okay. Suppose this is our new cell or old cell or parent cell. And within cell you have different organelles present, like nucleus. With nucleus we have our endoplasmic reticulum attached. Then we have Golgi body. Then mitochondria. Okay. From this cell we have to produce two new cells, exactly like the parent cell. So we have to produce two cells exactly of the same size with the same organelles, like this. Okay, in this same nucleus, same endoplasmic reticulum, then Golgi bodies, and mitochondria. So, before reaching this stage, there must be a stage where all these things must be getting duplicated. Okay? So, I am drawing again. All these things will be getting duplicated, and this cytoplasm, this empty space, this cytoplasm must be also growing. Because of that only we have equal size of cells formed. So I am drawing that intermediate phase also. Okay. Nucleus, endoplasmic reticulum, then Golgi bodies, and then mitochondria. So here you can see that these organelles have duplicated, and what has happened? This cytoplasm has also increased. It has grown. Okay. So from here till here, this intermediate stage is known as interphase, in which the cell is preparing for division. So this preparatory phase is known as what? It is known as interphase, which is also known as resting stage. So this is the interphase stage. Interphase. So in interphase, what is happening? Our cell organelles is being duplicated. Then the amount of DNA which is present inside the nucleus, suppose this is our DNA, it is also getting duplicated and passing on to the daughter cells. Okay, so here our DNA synthesis is happening. Then what next? Our cytoplasm is growing. So growth of cytoplasm. Okay, now, so from interphase we can see that two new cells, means two daughter cells are formed. And this, from here till here, this phase is actually the phase of cell division or mitosis or M phase. So this is our cell division stage or mitosis or M phase. Okay, now so what we saw that after the division new cells are formed, and that division is known as mitosis. Okay, then after mitosis again the cell will prepare for the next division. So that preparatory for the next division is known as interphase. Then again cell were dividing and again mitosis was happening. Then again interphase will happen. Then again mitosis, and this cycle will keep on repeating and repeating and repeating. Okay. So here one important point we need to note that this interphase state, it is divided into three phases, three smaller subphases, and that phases are G1 phase, S phase, G2 phase. Okay. During these phases only, our DNA will be getting synthesized, our cell organelles will be getting duplicated, cytoplasm must be increasing. So we will see in which phase these particular functions happen. Okay. So let's study interphase in detail. Interphase is divided into three subphases. Interphase. It is divided into three subphases, which is known as G1 phase, S phase and G2 phase. This G1 phase is also known as first growth phase. Always remember this. It is also known as first growth phase. Okay. This S phase is known as synthesis phase. I will be explaining why they are known as all these phases. Okay. It is known as synthesis phase. Then this G2 is known as second growth phase. So G1 is known as first growth phase because in G1 our initial first growth is happening, means our cytoplasm is increasing. Okay. This S phase is known as synthesis phase because in this our DNA is being synthesized, more amount of DNA is being synthesized. And this G2 phase is known as second growth phase because in G2 more amount of RNA and proteins are being formed and our cytoplasm is further increasing. Okay, so let's write all these points. So during G1 phase what happens? Some amount of RNA and proteins are formed or synthesized. Okay. Then next what happens? In G1 phase, mitochondria in all cells, both plants as well as animal cells divide. It is duplicated. Okay. Then chloroplast. These chloroplasts are only present in case of plant cells. So these chloroplasts also divide in plant cells. Okay, you may write here plant cell. Okay, now next, volume of cytoplasm increases. Okay, just learn these points. It happens during G1 phase. Okay, now in S phase, what is happening? More amount of DNA is synthesized. So more DNA is synthesized. Then what is happening? Our chromosome is getting duplicated. I will be explaining. So chromo get duplicated. Suppose this is our chromosome with a single chromatid in the newly formed cell. So during the interphase, what happens? This chromosome is duplicated and it forms the next chromatid. So now this chromosome, this single chromosome has two chromatids because DNA was located on this chromosome only. Okay, so our DNA has also duplicated. See, DNA has duplicated. Okay, and this chromosome, it has produced the second chromatid. So this chromosome has duplicated. Okay, more amount of DNA is synthesized and chromosome has been duplicated. Now in G2 second growth phase, what happens here? Again, more amount of proteins and RNA are being synthesized to aid in, means to help in cell division. Okay, so here again remember, both in G1 as well as G2, amount of RNA and proteins are synthesized. So more RNA and proteins are synthesized. Okay, you may note these points. These are very important points. Okay, now next I will be drawing the cell cycle, complete cell cycle. So let's draw. So this is a cell. Okay, and in cell, okay, I am representing cell cycle. So first we have our interphase. So interphase was divided into three phases: G1, S and G2. This is G1, this is S phase, and again this is G2 phase, and then we were having mitotic phase or M phase. So this is mitosis was happening next. So this cycle keeps on happening. From mitosis we have our interphase. From here till here is our interphase stage. Okay, so from mitosis interphase is happening. In interphase we have G1, then S phase, then G2, and from again G2 mitosis was taking place in which our cell was dividing into two cells. Now here one important point you need to remember that some cells, they do what? They leave this G1 stage and they do not divide further. So some cells, they exit G1 stage and do not divide further. Means if they are formed once, they will not divide during their entire lifetime. Okay, so example of that cells are our nerve cells, our neuron and the brain cells. They are formed at the time of birth only inside our mother's womb. Okay, so if they are formed again in the entire lifetime, they will not divide further. So the cells which the cells which exit the G1 stage and do not divide further, that stage is known as resting stage or R stage or G0 stage. And examples are nerve cells and brain cells. Okay, if they are formed, they won't divide. Okay, now let's see what happens in case of such cell. So these liver cells, they divide once every 1 to 2 years just to replace the lost and damaged cells. Okay, the skin surface cells. So this our skin surface, it loses its cells continuously. So to replace those cells, our skin surface cells, they divide continuously. Okay, to replace those damaged cells. Now meristematic cells. These meristematic cells are present at the tip of our stem and at the tip of our roots. So at the tip, this is the tip of the root and this is the tip of the stem. So they are present at the tip of the stem and roots, and they continuously divide throughout the life of plant. Okay, these cells are found in case of plants only, and they produce flowers and birds. Okay, all these structures. Now germinal cells, these germinal cells, they undergo meiosis. Okay, and they produce sex cells. So in humans, in case of humans, I am writing here, in case of humans, in males have testes and females have ovaries. So this testis was producing sperm and this ovary was producing egg or ova. Now in case of plants, okay, the germinal cells are anther in case of male, and again ovary in case of female flowering plant. So this anther produces sex cells which is known as pollen grains, and this ovary again produces egg. Okay, now next is uncontrolled division of cells. Suppose this is a cell, and instead of just dividing into two cells, this cell divide and forms uncontrolled mass of cells. The division is happening in an uncontrolled manner. You are not able to control the division at any stage. So this leads to formation of tumor. Tumor is just this large mass of cell, okay, which is formed as a result of uncontrolled division, and this tumor may lead to cancer. Okay, so I hope all these things about the cells are clear. Okay, now let's draw the interphase stage in reality. Okay, let's see how a interphase stage actually looks under a microscope. So I'm drawing the interphase stage in case of plants as well as animal cell. So first I'm drawing in case of animal cell. Animal cell only have cell membrane. This is the cell membrane. Okay, and in case of plants we have cell wall. Cell wall is the outer boundary. This is cell wall, and inner to it we have our cell membrane. Okay, plant cell are rectangular in shape. This is cell membrane. I am writing CM. This cell membrane is also known as plasma membrane. Okay, now next I am drawing is nucleus. Nucleus is the control center of the cell. It controls all the activities that happens inside the cell. So this is nucleus. Okay. And inside nucleus we have our chromatin fibers, okay, which condenses in the form of chromosomes. This chromatin fibers contains our DNA. So this is our chromatin fiber. Here also chromatin fiber. Now, inside our nucleus only we have nucleolus present. This nucleolus was indirectly helping in protein synthesis. So this is nucleolus. Okay, now then there is this structure called as centrosome, which is specifically present only in case of animal cells only, and it is absent in case of plants. Okay, so I am drawing this. Let's use black. So this centrosome, it consists of two centrioles. These two are centrioles, and here we have aster rays, and this whole structure is called centrosome. Okay, centrosome contains two small structures called centrioles. These two small structures that I have drawn are known as centrioles. So centrosome containing two centrioles. Okay, now both have this cytoplasm. This empty space is known as cytoplasm. So this is our animal cell. And this is our plant cell. This is how a cell under a interphase stage looks under a microscope. Okay, so you may note this point that you won't be able to see any visible change in this chromosome structure. Okay, suppose we know that our earth rotates around the sun, but are we able to see that change? Are we able to feel that change? No. Similarly, in case of cells also, this interphase stage under a microscope, it seems to be unchanged, as if nothing is happening, but in reality we know that DNA synthesis is taking place, cytoplasm is increasing, chromosome is getting duplicated. So that's why this interphase stage was also known as resting stage. So note this point that in interphase stage, no visible change in chromosome. structure. No visible change in chromosome structure, and this interphase stage is also known as resting stage. Okay, we have done everything about interphase stage. So this is the last thing for today's lecture. So let's summarize cell cycle and see the phases of cell division. Okay, so cell cycle was divided into two phases. Okay, interphase and M phase. M phase, which is known as division phase, which is actually the division phase. Okay, so this interphase stage was divided into G1, S and G2 stages. Now here we need to remember this important point whatever I am going to write. So this M phase, it happens in two phases. First, what happens? First, our nucleus divides. So first, division of nucleus takes place. Division of nucleus. Then after division of nucleus, division of cytoplasm takes place. So this division of nucleus is known as karyokinesis. Karyo means nucleus and kinesis means division. So division of nucleus. So this is known as karyokinesis. Okay, karyo means nucleus and kinesis means division. And this division of cytoplasm is known as cytokinesis. Cyto means again cytoplasm and kinesis means division. So division of cytoplasm. Now this division of nucleus, which is known as karyokinesis, it happens through four sub stages, which are prophase, metaphase, anaphase. From here I am writing here anaphase and telophase. In next lecture, we will study these four stages in details and we will be understanding it through diagram. Before ending this lecture, I really want to address one very important thing, which is journaling. So, this journaling is very important in our life. No matter in what stage of life you are, just start journaling. In journaling, you don't have to do so much. Just take a paper and a pen and whatever thoughts are going into your head, just write it down. Okay, I have myself been doing journaling since past 3 to 4 years and this has really helped in my life. So in journaling, what happens? Like, our mind has so many thoughts. Our mind is filled with different, both positive, negative, confusing thoughts, and as a result we feel irritated, we feel angry, we feel depressed, so many things. So the best way to deal with all these things is just writing these thoughts on a piece of paper. Okay, just give it a try and believe me, you will really feel good, because I have been doing. That's why I am telling you to also start doing this. Okay, and one tip that I really want to give is that please, if you are writing something that you feel that if it by mistake, if it must be read by anybody, you will be in a bad situation. So please make sure whatever you are writing, you are keeping it at a safer place so that it may not be read by anybody else. With my experience, I am telling this. Or you may do this that write down the negative thoughts which you are having and immediately just analyzing all the thoughts, then tearing that paper and just throwing it away. You may do this. So please just start this thing. It will really benefit you. So see you in the next lecture. Thank you so much.